Hey there, plant scientists. Today I'm diving into the world of Murashige and Skoog medium, or as it's commonly known, Mississippi media. If you're working in a plant science lab, chances are you've already crossed paths with this essential tool. So, let's take a closer look at how to safely and effectively use MS media in your experiments. First things first, what exactly is MS media? Developed in 1962 by Toshio Murashige and Folke Skoog, Mississippi media is a plant growth medium widely used for the cultivation of plant cell cultures. It's become the gold standard for plant scientists everywhere. Now, why is MS media so popular? Well, it's packed with all the essential nutrients that plants need to grow and thrive. From macronutrients like nitrogen, phosphorus, and potassium to micronutrients like iron and manganese, Mississippi media has it all. But before you start using MS media in your lab, there are a few important precautions to keep in mind. First, remember that MS media is for research and development use only. It's not meant for drug, household, or other uses. Always consult the Material Safety Data Sheet, or MSDS, for information on hazards and safe handling practices. When preparing MS media, make sure to wear appropriate personal protective equipment, or PPE. This includes gloves, lab coat, and safety goggles. You don't want any of those chemicals getting on your skin or in your eyes. Now let's talk about the preparation process. Start by dissolving the MS media powder in distilled water. Use a magnetic stirrer to ensure it's fully dissolved. Once dissolved, adjust the pH of the solution to around 5.8 using HCl or NaOH. This step is crucial for optimal plant growth. Next, sterilize the solution by autoclaving it at 121 degrees Celsius for about 15-20 minutes. Make sure to let it cool down before using it to avoid any damage to your plant tissues. When it comes to culturing your plant tissues, aseptic techniques are key. Work in a laminar flow hood to minimize contamination. Sterilize your instruments with ethanol or a flame, and always keep your workspace clean. One more tip, keep an eye on your cultures regularly. Look out for any signs of contamination, and if you spot any, act quickly to prevent it from spreading, and that's it. By following these guidelines, you'll be able to use MS media safely and effectively in your lab. Remember, the key to successful plant tissue culture is precision and care. Thanks for tuning in. If you found this video helpful, don't forget to give it a thumbs up and subscribe for more plant science tips and tricks. Happy culturing.
If your lab bench looks like a chemistry set, you're a true scientist. Let's make some MS media. First, gather your ingredients. Ammonium nitrate, 1,650 milligrams. Boric acid, 6.20 milligrams. Calcium chloride, 332.20 milligrams. Cobalt chloride hexahydrate, 0.025 milligrams. Cupric sulfate pentahydrate, 0.025 milligrams. Disodium EDTA dihydrate, 37.26 milligrams. Ferrous sulfate heptahydrate, 27.80 milligrams. Glycine, 2 milligrams. Magnesium sulfate, 180.70 milligrams. Manganese sulfate monohydrate, 16.90 milligrams. Myo-inositol, 100 milligrams. Nicotinic acid, 0.50 milligrams. Potassium iodide, 0.83 milligrams. Potassium nitrate, 1,900 milligrams. Potassium phosphate monobasic, 170 milligrams. Pyridoxine hydrochloride, 0.50 milligrams. Sodium molybdate dihydrate, 0.25 milligrams. Thiamine hydrochloride, 0.10 milligrams. Zinc sulfate heptahydrate, 8.60 milligrams. Mix 4.4 grams of this powder into 1 liter of water. And there you have it. MS media ready for your experiments. Keep exploring, scientists.
Want to keep your MS media effective? Here's what you need to know. All media preparations should be stored at 2 to 8 degrees Celsius. This keeps them stable and ready to use. For dry powder, store it in a desiccator. Why? Because MS powder is extremely hygroscopic. It loves to absorb moisture from the air. Any exposure to atmospheric moisture can ruin your media. Watch out for signs of deterioration. These include a color change, granulation, clumping, or particulate matter in the powder. If the powder doesn't dissolve properly, that's another red flag. A change in pH or worse, your media failing to promote growth means it's time for a new batch. Remember, proper storage is crucial. Keep it cool, keep it dry, and keep it effective. Don't let your hard work go to waste. Follow these tips and your MS media will stay in top shape. Good luck, researchers. Hey there, plant enthusiasts. Today we're diving into the fascinating world of plant tissue culture. It's like plant magic, growing plants not from seeds but from tiny pieces of existing plants. But before the magic happens, we need the right foundation, MS media. Think of MS media as the special plant food that fuels our tiny plant adventures. It's named after two super smart scientists, Murashige and Skoog, who created this awesome recipe. This media provides all the nutrients, vitamins and sugars our little plant buddies need to grow big and strong. Without MS media, our tiny plant pieces wouldn't stand a chance. It's the difference between a seed struggling in dry soil and one flourishing in nutrient-rich ground. So, are you ready to mix up some MS media and unleash your inner plant wizard? Let's go. Now, before we grab our lab coats and beakers, let's talk about temperature. It might seem like a small detail, but trust me, it's crucial for success. Our MS media, tucked away in its container, needs to be at room temperature before we even think about opening it. Why? Well, imagine taking a perfectly iced cake out of the freezer and immediately trying to frost it. It wouldn't be pretty, right? The same goes for our MS media. Cold temperatures can cause unwanted clumping and make it difficult to dissolve properly. So let's be patient and give it time to warm up naturally. Remember, in the world of plant tissue culture, a little patience goes a long way. All right, our MS media is at room temperature and we're ready to roll. But before we open that container, let's talk about using the entire contents. You see, Mississippi media is like that delicious loaf of bread that goes stale if you don't use it all. Once exposed to air, it can start to absorb moisture and lose its effectiveness. We want our plant babies to have the freshest food possible, right? So, unless the instructions say otherwise, plan to use the entire container. If you only need a small amount, consider dividing the powder into smaller portions before you start mixing. Remember, in science and in life, it's always a good idea to minimize waste. Section 4. Precipitation, a balancing act. Now let's talk about something called precipitation. No, not the rain cloud kind. In the world of chemistry, precipitation happens when a solid forms within a liquid. Sometimes, depending on the concentration of our MS media and the other ingredients we add, we might see some salts precipitate out. It's like adding too much sugar to your lemonade. It just won't dissolve properly. Don't worry though. A little precipitation is usually okay. We can often redissolve it later by gently heating our mixture. The key is to follow the instructions carefully and add things in the right order. Just like in baking, precision is key to a successful outcome. Section 5, Mixing Up Success, a step-by-step guide. Okay, plant enthusiasts, it's showtime. Let's mix up some MS media and create the perfect environment for our plant tissue culture adventures. Grab your trusty graduated cylinder and measure out about 90% of the final volume we need with tissue culture grade water. Remember, accuracy is key here, so double-check your measurements. Now slowly add our powdered MS media to the water while stirring constantly. This helps prevent clumping and ensures everything dissolves nicely. Think of it like making the perfect pancake batter. No lumps allowed. Don't let any precious MS media go to waste. Give that original container a good rinse with a little more water and add it to our mixture. Every bit counts. Depending on the specific needs of our plant friends, we might add some extra goodies to our media like hormones or vitamins. It's like giving our plants a special treat. Just like Goldilocks and the Three Bears, plants need their environment to be just right. We'll use a pH meter to adjust the acidity or alkalinity of our media to the perfect level for our plants. Add the remaining water to reach our final volume. We're almost there. If our recipe calls for a gelling agent like agar, we'll gently heat our mixture until it turns crystal clear. This creates a solid media for our plants to grow on. We can now dispense our media into containers before or after autoclaving, depending on our preference. And speaking of autoclaving, to prevent any unwanted microbes from crashing our plant party, we'll sterilize our media in a validated autoclave. This ensures a clean and healthy environment for our little green buddies to thrive. Section 6, Tips and Tricks for Happy Plants. Congratulations, plant wizards. We've successfully prepared our MS media. Now let's talk about some tips and tricks to ensure our plant tissue culture journey is a resounding success. First and foremost, always use high-quality ingredients and sterile techniques. Contamination is the enemy, and we want to give our plants the best chance of survival. Second, pay attention to your plants. Observe their growth and adjust the media composition or environmental conditions as needed. It's all about finding that sweet spot for optimal growth. Lastly, don't be afraid to experiment. Plant tissue culture is a fascinating field with endless possibilities. Try different plant varieties, media formulations, or even experiment with different lighting conditions. Remember, the journey is just as important as the destination, so embrace the learning process, be patient, and most importantly, have fun. After all, there's nothing quite as rewarding as watching new life sprout from a tiny piece of plant tissue. Happy culturing!